All right, well, let's go ahead and get started here. So I passed back the content checks. Number one looked excellent for the most part here. I didn't have any low scores, which was amazing. There was a lot of confusion, like covalent versus ionic bonds, right? Well, I don't have time to go over it in class today. I did post a really nice video up on Moodle that kind of walks through it slowly yet succinctly, so the video is only like 2 minutes long. And I also made a note that we will go over this for the exam review day, so you can make sure we work out that confusion. Anyways, let's get started with class. There's so few of you. I feel like I'm missing a lot, so it must have been a busy weekend, exhausting weekend. Maybe everybody got outside, enjoyed the weather, did a couple of hikes this weekend, which was nice. So we're going to get back into the swing of things, right, and start talking about cells. All right, so we're going to kind of give an overview of cells today and all of the organelles that are inside of cells. But I wanted to start by talking about the two major kinds of cells that we might find falling in those two categories of life. Right? It's either going to be a prokaryote or a eukaryote. Prokaryotes, they typically have really simple cell structure, right? They don't have a lot of organelles or membrane-bound compartments inside of them, and they don't have a nucleus. Though they do have what's called the nucleoid, it's just a region where you would find the DNA, but the DNA is not inside of another compartment, right? So they're relatively simple. Versus eukaryotes, right, which are probably the cells you're most familiar with, right? They have a nucleus that houses the DNA. They're generally much more complex. They contain many different kinds of organelles which are membrane bound. We're going to start by just reviewing prokaryotes because it's not really the focus of this class. I do want you to kind of know the difference. I did put this slide in here. I forgot about this, but I'm not going to review this slide. This slide is in here generally just for you to look at because I'm going to go over every aspect of this figure within the lecture today. So if we start with prokaryotic cells, like I said, they're relatively simple, right? And we can subdivide prokaryotes into two different types of organisms, bacteria or archaea cells. Right? The major difference between them is being that bacteria are very, very common, right? And they live in a lot of different environments. Most of them are not harmful to us, harmful to humans, but there are some species that cause disease. Right? Versus archaea cells, they're going to be about the same size as bacteria. They're still prokaryotes. They still have a relatively simple cell design. But we often are going to find them in really extreme environments, like the hot springs of Yellowstone or in the Arctic. Right? And they're much less common than bacteria. Just kind of overview of the structure. I don't expect you to know all of this, but I did want to point out that we do have our plasma membrane here. Right, just like we have in eukaryotic cells. We do have a region where the DNA is found, not the nucleus, we call it the nucleoid. We do have ribosomes, right? So some of them can have ribosomes, and they are an organelle. Right? And they can also have other features like this fly, which are these elongations off of our cell wall here that really allows these cells to kind of stick to each other and to other surfaces, and they could have some flagella, right? And that allows for movement within our cells, like a searing movement. That's all I'm going to say about prokaryotic cells. Right? The rest of the lecture is dedicated to eukaryotic cells, because they're just much more complex. Right? And that's what you probably are most familiar with because we're eukaryotic organisms. Right? So again, Eukaryotic cells. DNA is housed in the nucleus. The nucleus is membrane bound. Right? That is a big difference from the prokaryotes. They have organelles, which are kind of like these membrane-bound compartments. Right? And each organelle will carry out a specific function. And we're going to run down through a whole list of those organelles at the end of the lecture here. And they come in a bunch of different varieties. Right? The cells can be different between species. They can be different within a species themselves. So they might have these specialized cell types. Think about a red blood cell looks very different from a neuron, right, or a nervous system cell. Right. They can be very specialized and vary quite considerably. Right. Now, for your homework tonight, you'll really be diving into the difference between plants and animal cells, but I do want to point out that there are slight differences between them, though most of what we're talking about today is also going to apply to plant cells. Plants, they have this extra cell wall, which just provides it with some structure. It provides rigidity to that cell. They also have chloroplasts that will carry out photosynthesis. We'll elaborate that on later. All right. So I'm just going to immediately step back and have you think about what are those main differences between prokaryotic and eukaryotic cells, but also what are the similarities? How are they similar? I'll give you 30 minutes, and it's also a good chance for you to ask me some questions. Okay. I was just being jolly. Just like your conversation. Thank you. Thank you. There's so many different cells and prokaryotes. Yes, but not prokaryotes. And this next I get the sense that we've already talked about this, and maybe 3 minutes is a long time for this question. So I know there's only 5 seconds. So let's come back together and talk about this. What are some of the main differences between eukaryotes and prokaryotes? Yes, here. Yes, Yes, instead they have what's called that nucleoid. Yes, what's another difference? Yeah, it can be more abundant. Yep. Definitely there's a lot of different species of prokaryotes. Now, for something I specifically mentioned, but could be what's another difference? Yeah, they have a really simple structure because they lack a lot of organelles. Right? But how are they similar? They both have DNA, yes. What else do they both have? It surrounds the entire cell. Yeah, they have a cell membrane. Yes. Right. So there's those main differences and similarities. We will continue on talking about our eukaryotic cells here. And we're going to start by talking about each component of the cell. And we're going to start by talking about the cytosol and the cytoplasm. Right? They sound very similar. They are two different things. Right? So if I'm talking about the cytosol, I am talking about inside of the cell all the regions surrounding my organelles. Right? It's kind of like the fluid of the cell, you can think of it as. Right? It's very different from the cytoplasm, in which the cytoplasm contains everything inside of the cell, including all of the organelles. Right? So if I say I'm talking about the cytosol, I'm talking about the region surrounding those organelles. If I'm talking about the cytoplasm, I'm talking about everything inside of the cell. Right? Now, because the cytoplasm contains everything, we're going to break it down into some components. Right? So today we're really going to talk about the cytoskeleton and the endomembrane system. Right? The endomembrane system being most of the organelles inside of the cell. And then on Wednesday we will finish it off with the semi-autonomous organelles. Right? They function a little bit differently inside of cells. So let's start talking about the cytoskeleton. Right? So the cytoskeleton is like this network of filaments, right? So the definition of a filament is something that's long and thread-like. Right? We are talking about filaments that are built by proteins, right? So they're long, thread-like. You can kind of think of them as like strings and sticks of proteins, right? And we have different kinds because in biology, nothing is simple, right? There's so much variation. And so we have these microtubules, right? They tend to be these really big ones, right? They're long, they're hollow on the inside. Right? These microtubules, they are not going to stay in place, right? They can move. They have what's called dynamic instability. So they can shorten and they can also lengthen. Right? So they're not stable. That's different from these intermediate filaments. Right? Called an intermediate filament because it's an intermediate size between the largest and the smallest filaments. Right? They are relatively stable. Right? They are not going to move a whole lot. We typically see them as like these twisted or rope-like structures inside of our cells. Right? And then the last one was an actin filament. So an actin filament, they can also be called microfilaments, which is what this image here is calling it. They are the smallest, they're really long, they're really big. Right? Now, all of these work together to provide shape to our cell, but also movement inside of our cell. Right? So it's called a skeleton because it's kind of like the skeleton of the cell. Right? It provides that shape. Now, these filaments, right, because they can move, right, are going to have proteins associated with them to provide some of this movement. Mm-hmm. And so when we're moving things, we often need a source of energy. This energy is coming from our molecule ATP, which we will hear so much more about when we talk about cellular respiration. Mm-hmm. It typically has these regions associated with our motor proteins. So those regions might look a little different depending on different types of motor proteins. So in our our image here, we have 3 different types of motor proteins. Okay? They all have a head. Right? The head is the part that actually physically connects with these filaments. So we have the green heads up here, green head here as well. These are orange. Don't know why they changed the color for that image. Okay? They have a tail. Okay? So the tail is this long connection piece in here. And then they also have a hinge. And the hinge is kind of like the source of the movement. So a good analogy is if you're walking on the ground, right? The ground is going to be the filament, right? The head is going to be your feet, right? So it's physically attaching yourself to the ground. Okay? Now the legs would be the tail of the motor protein. So it's that long structured part. Okay? Typically will connect to any cargo that these proteins might be carrying. And your hip is the hinge. Right? So, and some of these work very much like if you would see somebody walking. Right? The hinge is moving those tails along, connecting and reconnecting the head on different parts of these filaments. Now, I have this video here that I think is super helpful in kind of understanding the cytoskeleton, but it's also really cute. It's about a 5-minute video, so we're going to go ahead and watch it in its entirety here. Let's see if I can't dim these front lights.
Our brain is made of billions of nerve cells, and they're all connected. If we take a closer look, a nerve cell seems to have antennas. Most of them are receivers of information, but only one is a transmitter, called the axon.
If this works for us.
This axon is connected to several receivers of other cells, forming a gigantic neural network, the brain. Meet John. John is a kinesin, a motor protein. He lives inside a nerve cell, and he has a proper job. To ensure that a brain cell does his job properly, it needs the continuous flow of building materials, proteins. They travel through the cell using the cytoskeleton. If you would compare a nerve cell with a city, the cytoskeleton inside the cell would be the roads, and the traveling proteins would be the traffic. These materials are towed by motors along the roads. And just as in real life, there are different kinds of motors and different kinds of roads. John's sole purpose in life is to deliver his cargo to a specific place in the axon. He takes the main roads, and he walks in just one direction only. John's job may seem easy, but it's not. He has to overcome a number of obstacles to ensure that the right amount of cargo arrives at the right place. To make the journey even more difficult, John is not alone. Other motor proteins ride along with his cargo. They haven't woken up yet, but that will happen soon. The journey starts in the center of the city, just like in the center of the cell. To enter the axon, John has to pass a place called the axon initial segment. In this segment, there are 2 kinds of roads. The main roads that John uses, called the microtubules, and a lot of little alleys called actin. And here, our brave motor protein meets his first challenge, because one of his sleeping travel companions, myosin, has woken up and starts to cling to the actin. And there are a lot of alleys. Only brute force can save John now. Fate strikes again. The other companion, Dineb, wakes up, and he can only walk in the opposite direction of John, resulting in a tug of war. But there can be only one. Along the axon in which John travels, there are places called synapses. Here, the axon connects to receivers of the other cells. Regulating proteins call the shots here. This traffic police makes sure that all passing traffic gets to the right destination. If John's cargo is needed in this synapse, he will be stopped and myosins take over his load. But today, John's cargo is safe. But what he does not know that his road is under construction just a few blocks away. In our nerve cells, the cytoskeleton is changing constantly. Roads are built, but are also broken down. Facing this kind of obstruction, John has to find a detour. John isn't the only motor protein on the road. There are many more. Our nerve cells need a smooth traffic flow in order to perform well. A traffic jam due to problems during the journey may ultimately result in brain disease. Understanding the challenges John faces could improve treatment or prevention. Finally, John arrives at his destination. He has fulfilled his destiny. But several other Johns are just getting started.
So I think that's a cute video that really demonstrates the complexity of the cytoskeleton. There's a lot going on. And while I don't expect you to know everything that's going on, I do kind of want you, oh, I clicked the wrong button. Oh no. I do want you to know kind of like those main differences, like the microtubules versus the actin filaments. We might have a brief pause here. Are there any questions? All right, yeah. That's a great question. I don't know the answer to that. I just know there's a lot of interactions going on. I'm assuming it has to do with wherever that cargo needs to go, or maybe the needs of the cell has changed and they have to switch where it's going. That would be my guess. I tried to turn the volume off so we wouldn't get any surprises, but it didn't work. I turned the whole thing off. That is on me. So these motor proteins, they have all these important jobs of carrying the cargo to where it needs to go. That video illustrated it in a neuron, right? But this happens in every single cell. Let's reconnect. It says I'm connected. Come on. Let's try that. Ah, there we go. All right. So we saw a really good example of a motor protein moving along the filament, but there are other types of movements as well. So we can have it where the motor protein doesn't actually move, but the filament instead will move. So in that example, it's like, if you think of my arm here as the motor protein, right? It's staying in place. And this other arm is the filament, right? It's going to attach, bend, and move the filament, reattach, and move the filament even further. All right, so in this case, the filament is the one doing the moving, not the motor protein. And even beyond that, there's this type of movement that's kind of like bending the filaments, where the filament isn't moving, the motor protein isn't moving, but the force enacted between the two is causing like a bending motion. And I think a really good example of this bending motion can be found in like flagella and cilia of cells. Right, so flagella, they normally could be long. They aid in a lot of movement of different types of cells through a fluid. Cilia, they're often really short and covering the entire surface of a cell, but they all involve that propagation of a bend. right? Similar to the motor proteins and the filaments both staying in place and initiating a bend instead. All right, so I have this group question here. I found this image online. I want you to explain to me this image. How is it correct? How is it inaccurate? All right? And it's all relating to this core cytoskeleton, which I think is very apt for the season, right? We're getting into that spooky season. All right, so take a moment, talk with your partner, wake up a little bit. I know it is Monday. Yeah, I know there's a lot of details of the cytoskeleton. I never make this back now. How's it going? I realized when I put it in full screen, it kind of cut off half the image, so. All right, so what are some examples in which this image is accurate? Yeah. Yeah, it's structure to the cell. Yeah, and it's just kind of hanging out, right? Which ways might this be accurate? Inaccurate, I should say. I saw you shaking your head. No. You want to elaborate?
It's not just like sitting around because it's always moving with every time.
Yeah. So unlike the skeleton that's just chilling, cytoskeleton is very active. Right? We have a lot of motor proteins either moving along the cytoskeleton or moving the cytoskeleton itself. Right? It's always building and under construction. Any additional questions? All right, well, let's go ahead and move on to our next slide then. So we talked about the cytoskeleton. Part of the cytosol is also the endomembrane system. So let's focus our attention on organelles now. So the endomembrane system is really all the organelles, pretty much all the organelles inside of a cell, and how they're all connected to one another. Right? might be physically connected, like they're physically touching, right? So a good example of this is we have our nucleus in purple here, and the endoplasmic reticulum is physically touching that nucleus. Right? Or they might be connected in that they work together, right? So maybe proteins are being transported between different organelles. Right? All these organelles, they make the cell work, right? They do everything from synthesizing to storage of proteins to cellular metabolism to breaking down different macromolecules, right? So these are the organelles that we're going to go over today, right? And I was really afraid we wouldn't have time to get through all of it. I think we're actually going to do it, right? So we're going to start with the big one, right? The nucleus, right? So the nucleus, it contains the DNA, right? The DNA is kind of the blueprint for protein synthesis. Right? And when it's time to synthesize these proteins, this DNA, which does not leave the nucleus, right, will create what's called messenger RNA, or mRNA, and that RNA will leave the nucleus and start describing to other parts of the cell how to make up proteins. Right? So on our nucleus here, right, while it does have this membrane-bound nucleus, there are these pores on the nuclear envelope in which that messenger RNA can leave and travel to our next organelle, which would be the ribosomes. Right? So the ribosomes, they take this messenger RNA, right, and actually decode it into a protein, right, through transcription and translation. Right? We'll get to that later in the semester here. Right? If you are a cell that has to make a lot of proteins, you will have a lot of ribosomes. Right? Likewise, if you're a cell that isn't so much involved in protein synthesis, right? Maybe you're a cell that could be specialized for storage of materials, right? You're not going to have as many of these ribosomes. Now, these ribosomes can just be suspended in the cytosol, right? In that fluid surrounding all of our organelles, or they can be attached to the endoplasmic reticulum, which I abbreviate on my slides as ER. Right? So the endoplasmic reticulum, like I said, ribosomes can be attached to it. And that kind of splits our endoplasmic reticulum into two different parts. We have what's called the rough ER and the smooth ER. So the rough ER is where the ribosomes are attached to the endoplasmic reticulum. You can see that in this cartoon image here. All these little yellow dots are the ribosomes, and they are attached. They're likely going to be attached closer to the nucleus than further away. Same here in this image, we see the green is the endoplasmic reticulum, and all those little red dots are the ribosomes that are attached. So this endoplasmic reticulum, it's this network of membranes, and it does a few different things, right? So when we have our proteins, or our ribosomes attached to the endoplasmic reticulum, it helps in protein synthesis. So essentially, the ribosomes will decode the information, the proteins will start getting made, and as they're being made, they're deposited inside that endoplasmic reticulum. The endoplasmic reticulum can also make more membrane for itself, right? So also helps to constantly keep replacing some of the membranes here. If we're looking at the smooth ER, which lacks the ribosomes, they're not making proteins, but they're still making some sort of molecules. And those molecules would be things like lipids, phospholipids, steroids, oils. Right? It can also help and store a lot of calcium ions. Now, I like this next image here because it kind of shows how the ribosome and the endoplasmic reticulum work together. So we have our ribosome here. It's attached to my endoplasmic reticulum in blue. As that protein is being made, right, it's being synthesized inside of the rough ER, it goes through all its stages of folding until it's either a fully fledged protein or pretty close to being a fully fledged protein. We might have a process, this is called a glycoprotein, because in this particular regular protein, we have added on some sugar chains to our amino acids. We're not going to go over that process, but that's why it's called a glycoprotein up here. Once that protein is ready to move on, it's going to go towards the edge of our endoplasmic reticulum, which will then kind of enclose the protein and bud off. Right? That protein will now be transported to wherever it needs to go next. Right? Now, sometimes where it needs to go next is the Golgi apparatus. Right? It has many different names: Golgi body, Golgi complex. Right? This is kind of similar to the endoplasmic reticulum in that it's made up of a lot of membranes, but these are stacks of flattened membrane-bound compartments. Right? And it's those vesicles, much like we saw on that last slide where it's budding off, right, that will transport materials to the Golgi apparatus as well as between all these different compartments of the Golgi apparatus. Okay. Now this organelle has a few different functions, and we're not going to go too deep into any one of them, right? But they can be involved in secretion of materials to the outside of the cell. And we can see that here we have this vesicle budding off. It's being transported to our plasma membrane where it opens up and the cargo is released outside of our cell. It can be part of processing proteins. Right? So I told you that the endoplasmic reticulum, right, once a protein is synthesized, it can be budded off. And sometimes those proteins aren't wholly complete, and it finishes up in the Golgi apparatus. And when I say finishing up, I don't mean adding more amino acids, but maybe different side chains or different carbohydrates or sugars being added on to make that protein function the way that it should. Right? So we're not adding more amino acids, we're just refining the protein. And finally, it involves protein sorting. Right? So there's a lot of different proteins in our bodies, and they contain a vast variety of functions. And the Golgi apparatus helps to sort them into categories and send them off to where they need to be. Now, if we don't need something in a cell, right, this is where lysosomes can play a role. Lysosomes, they contain enzymes, particularly the enzyme acid hydrolases. Right, it's a whole class of enzymes that perform hydrolysis. Right? So opposite of a dehydration reaction, hydrolysis, we're introducing that water molecule in order to break a bond. Right? And they go through the cell and they can start to degrade various different materials within our cells. Right? So they can break down proteins, lipids, nucleic acids, whole set of biomolecules. Very, very similar to peroxisomes. Right? So peroxisomes also catalyze chemical reactions using an enzyme, but the enzyme that it's using is different. So before the enzyme in a lysosome, right, used facilitated hydrolysis of a chemical reaction. Right? This enzyme, it's a class of enzymes called oxidases. Right? Instead of using water to facilitate the breaking of a bond, right, it eventually will produce, in the process of breaking a bond, hydrogen peroxide. Right? And again, they're breaking down a whole variety of organic materials. They are particularly important in liver cells because they can detoxify a lot of different molecules that might enter into our bodies. Right? And they are formed by budding off of the endoplasmic reticulum. Right? Lysosomes are formed by budding off of the Golgi apparatus. Very, very similar in their functions, but slightly different in what functions they specifically perform and where they originated from. Peroxisomes also tend to be a little smaller than lysosomes. All right, I think this is our last organelle that we're going to go over here today. Vacuoles. Right? So these are membrane-bound compartments. They can do a variety of things. In plants and animals, they're often used for storage. Right? So you can store whatever excess molecule we might need to store. In plants, they're used to store water. You can see a plant cell up top. It has this really, really large vacuole. Right? Versus in animals, that vacuole is going to be much smaller. In plants, they store a lot of water in their vacuoles. In other types of organisms, like algae, we have what's called a contractile vacuole. It will take up any excess water from the cell, and eventually when that vacuole is full, it will expel that water into the surrounding environment. So in algae, they live in very aqueous environments, and they can have such a thing as too much water, right? So they will take up any excess water and expel it for them. And in a lot of different protists, like this paramecium here, right, these vacuoles can actually help and degrade food materials. Right? So we might have food stored in our vacuoles. These vacuoles can, in conjunction with a lysosome that helps break down different materials, right, will degrade any food particles. Right? So they have a variety of functions depending on what species they're found in, what type of organism they're found in, plants versus animals, right? But mainly they're this membrane-bound compartment. All right, we are finishing up a little early, which is nice for a Monday. Okay. I do want to introduce this next content check. It's not due till next Monday, but I want to put it on your radar. You can start working on it, though we haven't really covered some of the concepts on it quite yet. And then homework for Wednesday. I got feedback that you all liked the video, so I'm saving the videos, but I'm doing something a little different for how you will submit. So it's not like a quiz, but it's really short answer to questions. Right. Any questions on anything so far? All right. Well, enjoy your extra 5 minutes here.